So in this video, we are going to talk about DNA markers. What are DNA markers? Different type of the markers which are used to identify the difference between individual and why they are important and the different ways of identifying this. So please make sure to like, share and subscribe my channel. So what is a DNA marker? A genetic marker or a DNA marker is a gene or the DNA sequence which you know the location, and based on that you identify the difference between individual. So suppose there is a variation between two individuals, or there is like the tests that are now done to check the relatedness between two individuals, like if the offspring, like if the father is the paternity test or the maternity test that are done these days are based on the DNA marker. So based on this you can identify the difference between individuals. So what are these markers? So there are basically three different type of markers which are used. So first can be biochemical marker, like based on the some proteins like the enzymes, how they are behaving, if there is mutation in one individual and there is no mutation in the other individual. So based on that you can identify the changes between two different individuals. And then the protein binding pattern, like if the protein is present or not present. So based on that also we can see if there are differences in individual or not. Another one is morphological. By looking at the features like the phenotype, we can observe the shape of the, suppose we are talking about the plants, shape and size and color. Based on that we can say if they are different or not. And the other one is molecular markers, which are important. So they can be used, like if you want to see the if you want to see the changes in the DNA sequence, you can use the PCR technique to amplify that sequence, and then based on different technique you can identify it. Or the hybridization, in which you transfer it to the another gel or like another membrane to see the differences between individual, like the hybridization, and then you probe for some antibodies or some, sorry, probe for the DNA. So this is also one of the technique which is used. So we will talk about all of them in brief. So what is the morphological markers? So as we have talked about, you can, by looking at the features like the shape, size and colors of various plants part, you can see, you can say if they are related or not. And they can be used for like the identification, like if they belong to same variety or if there are some differences in the differences or not, just by looking. So there are some advantages of this technique. Obviously it's available all the time and it does not require much knowledge about the things, like it requires that, like you just need to see. So there is no proper, like you need to do PCR to identify. It's like a visual technique and it's a direct measure. But there are advantages sometimes, like by looking at the morphological features you cannot say if the genotype is the same or not, right? So this is one of the important thing, like it's a phenotypic marker, it's not genotypic marker. So suppose like you need to be expert, like the shape is round or not, or if it's wrinkled, so you need to be expert on that thing. And as we know, like most of the characters are also influenced by environment. So if we see the difference, but it doesn't mean that the difference is mostly genotypic, it can be from the environment as well. And obviously there are limited numbers of the marker, like as we have talked about, which can we can see, but the others we cannot see. So this is a, it has advantage that it's available rapidly, like you can do it very easily. But then the major disadvantage is you cannot look at the genotype. And then comes the biochemical markers. So, as the name suggests, biochemical means something which can be biochemically related, like the protein or amino acid binding pattern. So, as you know, like the electrophoretic techniques are basically used for identification. Like you can run the STS gel based on the like the enzyme activity. Like if you see the peroxidase acid, phosphatic acid, right, they convert the substrate to the product into a different, remove some like esterase, remove the ester group. So based on this we can see the binding pattern if they are available to do that or not, like basically the protein. So in biochemical markers we are not looking at the gene level, we are looking at the protein level. So it's like it's require very simple technique. You just need to look at the protein and then assess the differences. And then it obviously, because in the morphological we are just seeing the morphological features, by adding this we are adding another layer which is the protein. But obviously there are disadvantages, like sometimes there are changes in the gene level but the protein remains the same, and so we don't know. And sometimes the change, like there are changes in the non-coding region which we cannot identify by this, because the coding region is the same, so the protein will form. And then obviously it also subject to environmental condition, and there are limited these enzyme which can be looked at. Other enzyme, other sequence, gene sequence mostly like does not have this enzyme which we can clearly see. So the biochemical markers are also good, but obviously they also have the limitation. And now we go to the molecular markers, which are very important, are mostly focused on the gene sequence, and they are mostly used to identify the difference between individuals. So as we talk about, molecular markers are basically the DNA sequence which identify the changes in the DNA or the protein sequence within the chromosomal region. And there are two type of marker, like the gene products. So suppose we know a gene and we are looking at that product, whether it's acting as a enzyme, allozyme, or so we are looking at the protein. And the another one is DNA marker, which is used for the genetic stock identification, like the transfer of character, the hereditary. It's basically the DNA marker which are used. So now we are going to talk about DNA markers and how they can help us, right? So first is, we are directly working with the genotype. So we are looking in the morphological one, we were looking at the phenotype, but the phenotype sometimes can be influenced by environment, and you don't actually see the difference in the genes, and sometimes the phenotype is the same but you have difference in the genotype. So with molecular markers we are directly working with the genotype, and we can analyze the effect of genotype. So suppose we know, like, okay, this individual, like these two individuals has different on this gene, and then we can look how in the phenotype they are different. So we can go from genotype to the phenotype, and then we can look obviously at the genotypic level, which is very important to look at the genotype, because the most of the information that we have is on the protein level. So, but it's very important to look at the genotypic level, how is it happening. So now we are going to talk about different techniques which are used to identify these molecular markers. So first is this restriction fragment length polymorphism. So what does that mean? Is restriction fragment. So as the name suggests, these techniques work with the restriction enzyme. It actually check if between the two individuals, if we cut with the restriction enzyme, with the same restriction enzyme, how the pattern we get. And so as you can see here, there is one sequence which is for the normal condition, and then suppose there is a disease in which this mutation occur. So when we cut them with the same restriction enzyme, so as you can see here, here it cut with the MST1, right? And in the normal condition there are three sites which where MST1 can cut, but in the disease condition there are only two, right? So when we run this the gel electrophoresis, and then we transfer it to the southern blot. So this is the hybridization technique, right? So we transfer it to the southern blot, and then we probe for this bands only which have the restriction site. So here you can see in the normal condition we have two bands, but in the disease condition we have only one, because here we get only one band, but here we have two bands of different size. So based on the restriction fragment polymorphism, like based on the different restriction enzymes, we can say if there are differences in the genotype or not. And then we can go in deeper, where are the differences, how it relates to the disease condition or not. So this is one of the technique which is used to identify the difference between individuals. So next is RAPD, or random amplified polymorphic DNA. So in this technique, there is a random amplification of the polymorphic DNA using a different sets of primer which are not very specific but are random primers. So suppose we have a DNA template A and DNA template B, and then we put a 10, like so many primers in this condition, and wherever there is any similarity the primers will bind, and then we do the PCR amplification. So if there is difference between these two individual or between these two DNA sequences, we will see different bands, right? So here you can see there are two sets of primer, forward and reverse primer binding. So we will see two sets of DNA bands, but in the DNA template B we only see one band. So here we, based on the, so this is, in this time it's not the specific DNA specific primers that we are using, it's the random primers that we put on, and then we see the differences. So this is very important, like just to check if there are differences or not, and then we can go specifically where are these differences and how can it relate to the phenotype, and if there are related to some disease or not. And the another one that we are going to talk about is the amplified fragment length polymorphism. So it's a combination of the RFLP, restriction fragment, and the random amplification technique. So it uses both the restriction enzyme, restriction fragments, and then the amplification with random primer, like, and then going very specifically. So here suppose we start with the genomic DNA, and then as we do in the RFLP we digest them with some specific enzyme. So here suppose we digest it with the two specific enzyme or directly with the TAC1, and then we get this sequence, right? After digestion, suppose it cuts here and here, and then to identify the sequence, what we do is we add the adapters, right? So here is a sequence after the digestion, so we ligate it with this TAC1 adapter. So here you can see the TAC1 adapter, and now we know the sequence of this adapter, okay? And then we do the amplification by PCR using the primers for which we know the sequence of this adapter, and then we take only one single selective nucleotide from this sequence. So suppose there are individuals in which there are differences in the first sequence from here. So here you can see from my left hand side we are taking only one specific nucleotide here. So this is the amplification with unlabeled primers, and then we go ahead and then we again do another amplification, but this time we can take more than one. So either we can take 2 or 3 selective nucleotides, and then we do the amplification. So this is the combination, like we are adding more complexity. We are adding restriction enzyme, but then sometimes the restriction enzyme is the same, like the site where it's bind is more or less the same but not exactly, right? And then we are again, after moving further, and then we are specifying different primers with either one nucleotide specificity, and then moving to 2 or 3. So this is very specific, and it tells you the difference between the individuals, like very specifically. So this is an important technique which is used. And now we move to the another one, which is a microsatellite markers. So this is basically so there are basically some non-coding DNA region in which there is repeats of this CA repeats. So, and in some individual these repeats can be 4, 5, 6, 7. So this technique is basically used to identify this kind of repeats, if there are differences or not. So suppose we have two DNA sets or two alleles. In one of them there are 4 repeats, in another there are 8 repeats. So when we do the amplification with primers. So based on the repeats, we will have different bases, right? And then when we run them, the less the base, the more it goes. Like the smaller the size, the more faster it will go, and the larger the size, it will be on the top. So based on this, we can identify how many repeats are there, and if there are in difference between two individuals or not. So I hope it's clear, and if you have any doubts, please let me know in the comment section, and please make sure to like, share and subscribe my channel.